So welcome to the 7th session in this mass spec course, and in this session we're going to be going through a basic introduction to tandem mass spectrometry. Earlier in these lectures, we revised the concept of electron ionization. Electron ionization is an example of a kind of hard ionization, where the ionization process leads to fragmentation. On the many occasions when you can't really see the molecular ion in EI, it makes it difficult or impossible to determine the molecular weight and formula of the component you're analysing. And it's because of this that most modern ionization sources used in biotechnology or medical research are designed to be soft ionization sources that mostly produce molecular type ions. Examples of this kind of soft ionization source are MALDI and electrospray, and we'll cover electrospray in the next session. For soft ionization sources, there are some pros and cons to their use. On the pros side, because they're mostly showing the molecular type ions, it's easier to determine the molecular weight and chemical formula. And because you're not producing multiple fragments, just the molecular type ions, that means the spectra are much simpler than they would be otherwise. However, there is a con to this soft ionization, and that is that because you're not seeing significant fragmentation, you actually get much less structural information about the molecule you're looking at. And this structural information is vital in biochemical or pharmaceutical analysis, because it's the only way you can distinguish between multiple possible isomers that might all be in the system you're looking at. So if we still need to use soft ionization in order to get the molecular mass, how do we get the structural information back? And the answer to that little conundrum is a process called tandem mass spectrometry. So in tandem mass spectrometry, we take that molecular type ion that we've made from our soft ionization source, we isolate it in the mass spectrum, and we deliberately fragment it. Molecules don't fragment completely randomly, they'll fragment in ways that make sense based on the structure of the molecule. So by taking a mass spectrum of the fragmentation products, and we'll call them daughter or product ions, we gain more information about the structure of the original ion. And then combining that structural information with the molecular weight that we got from the mass of the parent ion, and from the chromatographic retention time, because this method's usually used after chromatography, we can be really confident about the identity of the parent ion. As I alluded earlier, tandem mass spectrometry is often given different names. Sometimes it's called Tandem MS, or MS-MS, or MS squared, or MS to the 2. And all of these mean the same process, isolating a parent ion, fragmenting it, and then analysing some or all of the fragment ions. But in tandem mass spec, you're not limited to one round of fragmentation. So if your system allows it, you can isolate one of the fragments from your first fragmentation, fragment that fragment, and look at the product ions of that. And on ion trap systems, you can repeat that process again and again, providing you've got enough ions to detect. And this process can be called MS-MS-MS, or MS to the 3, or even MS to the n if there's multiple different stages. MS to the n is an extremely powerful technique, and it's been fundamental in our ability to characterize complex biochemicals such as metabolites, lipids, and pharmaceutical products. But given that the whole point of a soft ionization source is that it generates ions that don't spontaneously fragment, how do we fragment these ions when we choose to do so? Well, clearly we've got to put some energy into the ion in order to get it over the fragmentation energy barrier. So all the different fragmentation techniques used in tandem mass spectrometry are just different methods of depositing energy into ions. The most commonly used method are the collisionally induced methods, so that's collisionally induced dissociation or collisionally activated dissociation. And these account for by far the largest proportion of all tandem MS experiments. Next, there's a class of tandem MS methods that all use electrons. So there's electron capture dissociation, electron transfer dissociation, and electron-induced dissociation. All of these processes are different, but because they all have a similar structure of acronym that goes electron something dissociation, they're often grouped under the general heading EXD. And all of the electron-based fragmentation methods operate by forming a radical somewhere on the structure of the molecule you're trying to fragment. And the last class of these fairly common fragmentation techniques are the laser methods. Infrared multiphoton dissociation uses an infrared laser to put heat into the ions you're trying to fragment. And it often produces fragments that are quite similar to those that you'd get from the collisional heating methods. The last method shown here is one that's growing in importance, and this is ultraviolet photodissociation. In this method, the ultraviolet photons each carry enough energy to generate photoelectrons from the molecule you're trying to fragment. And of course, having knocked an electron out of that structure, what's left behind is a radical, and that can then go on to spontaneously fragment. In collisionally induced dissociation, we collide our ions with background gas molecules, and this has the effect of heating up the ions, causing the weakest bonds to break. Normally, argon or nitrogen is used as the collision gas in these experiments. In the most common way that CID is implemented, each collision alone provides only a small amount of energy, so many collisions are needed to raise the internal energy of the molecule high enough to induce fragmentation. But you don't necessarily see in the mass spectrum all the fragments that are created by this process. The fragments that you see, yes, will result from the breaking of weak bonds, but you only see the stable ions, those that exist long enough to be detected at the end of the experiment. The stability of ions depends on their structure and whether or not they have some resonance stabilization within them. Unstable ions, if they're created, quickly go on to decay into smaller fragments. And that process of fragmentation can keep happening until a stable fragment is formed. Some fragments that are created are not completely stable. Some proportion of them will last long enough to be detected, while some other fraction will decay onto smaller fragments. So the relative intensities of the fragment peaks you see in your final mass spectrum depends on two processes. Firstly, the probability of formation of that ion, but also the stability of that ion, meaning that it doesn't then quickly go on to fragment to something smaller. So let's look at an example of a time when MSMS is necessary to distinguish two compounds. Morphine and hydromorphone are structural isomers of each other, differing only in the location of a double bond. So that means their molecular class ions will have exactly the same mass. So irrespective of how good the mass accuracy or resolution of our mass analyzer is, we'll never be able to tell them apart by mass spectrometry alone. But if we isolate the ions containing the complete molecule and then subject them to fragmentation inside a mass spectrometer, the pattern of fragments that's revealed does allow us to differentiate between the two products. When we fragment morphine, we get major CID fragments at 201, 229, and 268. But when we fragment hydromorphone, we see the major fragments at 185, 229, and 227. In these MSMS spectra, it's easy for us to distinguish morphine from hydromorphone because the fragment ions produced after fragmentation have different masses. For some compounds, for example stereoisomers, the differences even in the Tandem MS spectra can be even more subtle. Very occasionally you'll find molecules where even the fragments have the same masses, and the only way you can distinguish them are subtle differences in the relative intensities of those fragments. One other thing you might have noticed in this data is that in the tandem mass spectra there are no carbon-13 peaks, but in the original mass spectra there are. The reason for this is that when the parent ions were isolated, only the monoisotopic peaks were selected. Therefore, there are no heavier isotopes at all in any of the fragments. So it's worth remembering that when you're selecting ions for isolation, if you don't isolate the entire isotopic distribution, then the isotopic distribution in the fragments will be affected by this. Now, having looked very quickly at the basics of tandem mass spectrometry, it's time to look at the 4 standard tandem mass spectrometry experiments that are commonly run. These are the product scan, the precursor scan, the neutral loss scan, and selected or multiple reaction monitoring. All of these experiments were developed on the assumption that in most samples there will be a mixture of components to be analysed. And what's the normal method of taking a mixed sample and then presenting only one component at a time to a mass spectrometer? Well, that's chromatography. So these methods strongly reflect the fact that most tandem mass spectrometry is done on LC-MS systems, where liquid chromatography is separating the sample before MS analysis. And in the next session, we'll look how the time relationship between LC and MS was the driving force behind the development of these 4 standard scans. But for now, let's go through the 4 of them in more detail. The product scan is the most basic experiment. When people describe tandem mass spectrometry, normally what they're describing is a product scan, where you select an ion of a known mass, that selected ion is fragmented, and then the system produces a mass spectrum of all the fragments from that selected ion. The Tandem MS data we looked at for morphine and hydromorphone a couple of slides ago were produced by product scans. So experimentally, why and when would you undertake a product scan? Well, the aim of this experiment is to help you find all the fragments of a given ion. And then there are 3 main things you might do with that information. The identification of the fragment ions might give you enough information to structurally characterize your original ion. Or possibly you're going to compare the fragments from this ion with other ions that you might confuse it with in order to identify either the most abundant fragments that will give you the most sensitivity if you're trying to detect this molecule, or the most informative fragments that will most uniquely identify this molecule. And then we'll come back to how we use the information about the most abundant or most informative fragments when we look at the selected reaction monitoring mode later. The second standard tandem mass spectrometry experiment is known as the Precursor Scan. So imagine you have a sample containing many components. But within that mixture, you might only be interested in a certain subset of the chemicals that may belong to a particular chemical class. So a precursor scan is intended to allow you to look through all the data from that sample and find all the parent ions that create a specific fragment mass. In a tandem mass spectrometry experiment, there are 2 distinct stages of mass analysis, with a fragmentation stage in between. In a precursor scan mass spectrometry experiment, the second mass analyzer is set to look for only one fragment mass, while the first mass analyzer scans through all possible ion masses. Because the mass analyzers are in series, you only see a signal on the detector when you have an initial ion which fragments to produce the fragment you're interested in, and then you can correlate the time of that detection to the mass of the original ion. So having explained to you what a precursor scan is, now you're probably wondering why on earth would we develop such an experiment? Well, the aim of a precursor scan is to help us identify compounds with common structural motifs, such as members of the same chemical class. But in tandem mass spectrometry, molecules don't fragment randomly, they fragment according to rules based on their structure. So a precursor scan can help you find members of a chemical class which all lose the same ion upon fragmentation. And a great example of this is phosphopeptides. Phosphorylation is one way that your body turns the action of proteins on or off. So being able to identify which proteins are phosphorylated in a given sample can provide really important information about the biochemical environment that sample was taken from. For example, giving information about how a disease works, or how the body is responding to an injury. The normal method of analysing such samples is known as bottom-up proteomics. So in this case, you might take a complex sample like a piece of tissue or blood plasma, use an enzyme to digest all the proteins in it down to peptides, thousands and thousands of peptides, and then analyse these in a tandem mass spectrometry run. But because we're interested in which proteins are phosphorylated, what we want is an easy way to identify which of the many, many thousands of peptides in this sample have been phosphorylated. Luckily, when fragmented by CID, all phosphopeptides will tend to produce a common phosphate fragment at MZ79, So we can use a precursor scan where the second mass analyzer is set to 79 to identify all the parent ions that can produce this fragment, and these are most likely to be phosphopeptides. Then, having identified the chromatographic retention time of these phosphopeptides, then you could go on to analyze them using a different method. The neutral loss scan is similar to the precursor scan in that you're using it to try and identify members of a restricted chemical class, only this time the signature of that class is that they will consistently lose a particular neutral mass during fragmentation. So just like the precursor scan, it'll help you identify compounds with common structural motifs. And an example of this would be looking for glucuronides in a sample. On the right of the screen, you can see 3 examples of different glucuronide compounds. As with other members of the class, during collisionally induced dissociation, these will all lose the glucuronic acid moiety that's attached to the end of the ring structure. Because we know these compounds will all tend to lose glucuronic acid upon fragmentation, that means we're always looking for a fragment which weighs 176 Daltons less than the parent ion. So to find glucuronides in a sample, using Tandem MS, we would get the first mass analyzer to scan across all masses, and then set the second mass analyzer to track the first mass analyzer, but with the mass of the second analyzer always being 176 Daltons less than the set mass of the first mass analyzer. That way we'll only get a signal on the detector for an ion which will lose 176 Daltons during the fragmentation stage. The last of the standard scans we'll look at in this session is selected reaction monitoring. This method is used to look for specific molecules in a complex mixture. And in fact, selected reaction monitoring is the industry standard method of quantifying and confirming the presence of specific molecules in complex mixtures. In this technique, the first mass analyzer is set to constantly isolate only a single parent ion mass, that being the molecular ion of the molecule you're looking for, while the second mass analyzer is always set to look for a specific fragment that is known to be formed by tandem mass spectrometry from the parent ion you're looking for. And why is selected reaction monitoring used? Well, it's the most sensitive and selective method we have. Correlation of a specific fragment ion coming from a specific parent ion at a specific chromatographic retention time gives you very high confidence that you've correctly identified the molecule you were looking for. Additionally, the tandem mass spec method of looking for a specific fragment coming from a specific parent removes an awful lot of the background chemical noise from the signal, therefore the quantification is the highest possible quality by this method. To increase the specificity even more, sometimes a related method called multiple reaction monitoring is used, and that's where you have the system look for more than one specific fragment coming from the parent ion of interest. Of course, for most of a chromatographic run, your molecules of interest are not eluting from the chromatograph. So it's not sensible to have your mass spectrometer try to look for a molecule you know shouldn't be there at that time. So often these are turned on intelligently. So for example, a selected reaction monitoring method may be turned on in the tandem mass spectrometry part of your system at the correct retention time for that product to elute. Or alternatively, your system can look to see if a particular mass corresponding to the parent mass appears, and if it does, then turn on the MS for that ion. So to recap, there's generally accepted to be 4 standard methods of tandem mass spectrometry experiment. In the product scan, you're trying to analyze all the fragment ions coming from a specific parent ion mass. In the precursor scan, you're trying to find all the parent ions that can create a specific fragment ion. In the neutral loss scan, you're trying to find all parent ions that can lose a specific neutral fragment. And finally, in the selected reaction monitoring mode, you're asking the question, does a specific parent ion exist? And if it does it produce a specific fragment ion? And also with the right chromatographic retention time. The graphic you can see on the right summarizes this information, and it's a modification from the figure in the paper you can see linked underneath. Mainly because it always made more sense to me if the figure had been this way round, but it's the other way round in the paper. There are two ways of using tandem mass spectrometry that are also worth mentioning in this session. These are data-dependent and data-independent tandem mass spectrometry. These are often abbreviated to DDA and DIA. We'll start by looking at data-dependent MS-MS. This is called data-dependent analysis because the selection of which masses to fragment and analyze by tandem mass spectrometry is made by the instrument and is dependent on the data in the mass spectrum that just precedes it. And in practice, this is how it works. It's normally used on samples which are being separated first by liquid chromatography before tandem mass spectrometry. So all the different compounds in the sample elute out of the LC column at slightly different times. Therefore, what is seen by the mass spectrometer also changes all the time. So in a data-dependent MS-MS run, the first thing the mass spectrometer will do is to record a mass spectrum with no fragmentation. Then it will detect the peaks in this mass spectrum and select the top 5 to 50 ones of those peaks for greater analysis. Then, in the next mass spectrum, it will isolate the most intense of those parent-ion masses, fragment it, and collect a product ion scan of the fragments so that it can search that against a library in order to try to identify that component. Then it goes on to the next ion in its list, fragments that, generates a product ion scan and searches that against the library, and so on. Once it's exhausted that list of parent ions, it collects another mass spectrum in order to generate the next list of parent ions to undergo tandem mass spectrometry. And then it repeats this cycle of collecting a mass spectrum and then a series of tandem mass spectra of different components until it gets to the end of the chromatographic run. There's one key point about the DDA process that I want to go into in a little bit more detail. I just described how, in an LC tandem mass spectrometry DDA run, the mass spectrometer repeats a cycle where it collects a mass spectrum, detects the peaks in that mass spectrum, and then undertakes tandem mass spectrometry on the top 5 to 50 peaks. But why is DDA limited to looking at only the top 5 to 50 peaks? Why not look at all the peaks in the mass spectrum? And the answer to that is simply time. Remember that in DDA the mass spectrometer is detecting peaks as they elute from the LC column, and LC peaks are only 10 to 15 seconds wide. Each tandem mass spectrum takes time to collect, so you're limited to the number that you can run on an LC timescale. If you try to detect too many, by the time the instrument gets on to isolating that particular parent ion, the compound that refers to may no longer be eluting from the LC column, so you won't see anything at all. You've missed the peak. So the number of tandem mass spectra that you can collect after every mass spectrum depends on the rate at which your instrument can undertake tandem mass spectrometry. The alternative to data-dependent analysis is data-independent analysis, and this is normally also done after LC separation. And it's called data-independent mass spectrometry because the selection of which masses to fragment is independent of the data in the preceding mass spectrum. There are a very large number of different DIA methods, and so you can't really say there's a standard approach. And indeed there's so many different ones that we can't possibly go through them all in detail in this module. But there are some common features. In data-independent analysis, as in DDA, mass spectra commonly alternate with one or more tandem mass spectra. But in DIA, when tandem mass spectra are collected, there's either no parent ion selection, meaning that all the ions passing through the instrument at any one time are fragmented all at once, or at least a very broad range of masses is selected for fragmentation, rather than trying to select only a single component. The benefit of this approach is that it overcomes one of the weaknesses in data-dependent analysis. Remember that in data-dependent analysis, the instrument selected which ions to fragment because they were the most intense peaks in the preceding mass spectrum. But of course this means that in DDA only the most abundant components in a sample get analysed, and lower abundance components get ignored. Data-independent analysis overcomes this issue because in DIA the system fragments everything going through the instrument at a given time, independently of whether or not there's an intense parent ion present. So in DIA you see the fragments even from the low abundance components in your sample. But of course there's a downside to this approach. In data-dependent analysis, the system was selecting a single component for fragmentation, so the instrument knows the mass of the parent ion that's responsible for any given list of fragments. And then when it searches the libraries of fragment spectra to try and match the spectrum it's seeing, it can discount any potential matches that must have come from ions with the wrong molecular weight, and this greatly cuts down the list of possibilities. But in data-independent analysis, although the instrument knows what masses were going through the instrument at any time, it can't directly correlate exactly which parent ions relate to which fragment masses, so it can't do this clever cutting down of the possibilities anymore. And it's for this reason that very clever software algorithms must be used to process DIA data. Most of the tandem mass spectra collected in a DIA run will contain fragments from multiple components, and therefore the software needs to find some way of deconvolving which fragments came from which original parent ions in order that it can build virtual fragmentation spectra to search against libraries. And remember that DIA is analysing a far larger number of components in your sample than DDA because it's not limited only to the most abundant ones. So we'll now go through two different data-independent analysis methods. These are PAcIFIC and MS. PAcIFIC stands for Precursor Acquisition Independent from Ion Count, and it was developed in the Goodlet lab in the late 2000s. In PAcIFIC, a tandem mass spectrum is required at every mass to charge value, whether or not a precursor ion is observed. In fact, no survey mass spectra are actually recorded in this technique at all. Now it's important to remember that in any tandem mass spectrometry experiment, you're limited to the number of fragmentations that you can record per cycle by the LC timescale. But the hallmark of PAcIFIC is that it uses a relatively narrow isolation window before tandem mass spectrometry. This makes it easier to relate the fragment ions back to an original parent ion mass, but it means you need a large number of windows to cover the full mass range. So how does PAcIFIC do it? Because you can't record hundreds of different tandem mass spectra in a cycle on an LC timescale. Well, the PAcIFIC method limits itself to 10 different tandem mass spectra per cycle. But because the isolation width for each one is narrow, only 2.5 MZ wide, that means it only covers a 15 mass to charge range in every cycle. And then this is repeated for the duration of the LC run. Of course, if you have a sample as complicated as blood plasma or a tissue homogenate, is a 15 Dalton mass range really that useful? And of course it's not, but this 15 MZ range is the maximum you can cover using relatively narrow overlapping windows on an LC timescale. But PAcIFIC overcomes this limitation by looking at a different 15 MZ range across multiple repeat LC runs of the same sample. In the example they provide in the initial paper, it takes 67 LC runs to cover the full mass range. In the first LC run, they looked at masses from 400 to 415, and in the next one 415 to 430, and so on until after 67 injections they'd covered the full mass range. The downside of this approach is that such in-depth analysis inevitably takes a long time. In the example in the paper, it takes over 4 days. But the benefit is that you know the fragment ions must have come from a parent ion within a relatively narrow mass range. And that means it's relatively easy to search this admittedly enormous dataset against normal libraries using conventional software. I've added a link to the PAcIFIC paper here at the top, so my recommendation is read this for yourselves to make sure you understand exactly how the process works. I previously showed this 2D view that summarizes the relationship between parent ion and fragment masses. So on this we can see where the product scan, neutral loss scan, precursor scan and SRM plot on the plane. And now we can add the PAcIFIC scan to this view. So for a single injection in PAcIFIC, you record 10 contiguous product ion scans covering a relatively narrow mass range. And then across multiple repeat LC injections, you move that isolation range and the 10 underlying product scans until they cover the complete mass range of interest. And in that way, your final dataset contains a tandem mass spectrum of every possible mass across your mass range, whether or not you would have been able to detect a parent ion there in a normal mass spectrum to begin with. If the hallmark of PAcIFIC is that a relatively narrow isolation window was used to collect product ion data, then the opposite is true of MS2E, because in this method there is no isolation window. All the ions that are travelling through the mass analyzer at a given time are fragmented all at once, and their fragments shown together. The MS2E concept was developed by a combination of Waters, Imperial College and AstraZeneca in about 2006. As with PAcIFIC, I've added a link to this paper up at the top of the screen. The basis of MS2E is as follows. During the full length of an LC run, 2 different types of mass spectrum are recorded alternately. The first type is just a simple mass spectrum, so the collision energy is turned right down so there's no fragmentation before the ions get into the final mass analyzer. Then, after every mass spectrum is recorded, a tandem mass spectrum is recorded, where the collision energy is turned on to cause fragmentation, but where the isolation window is set to the full mass range, so that there's no selection of any specific parent ion. That means this fragmentation spectrum shows all the fragment ions coming from all the precursor ions that were eluting from the LC column at a given retention time. And then the system records another mass spectrum, and then another tandem mass spectrum, then a mass spectrum, a tandem mass spectrum, and so on until the end of the LC run. But given this concept, how can you identify which fragments came from which original parent ion? Well, this relationship can be worked out in software because the mass spectra and tandem mass spectra are being recorded at a high enough rate that you can really see the shape of the chromatographic peaks as the components elute from the column. So unsurprisingly, you expect the fragments from a particular compound to change in intensity following the same trend set by the precursor ion of that same compound. So the software trawls back through the huge MS2E dataset to group together fragments and precursors that show the same retention time and the same chromatographic profile. And then these can be combined into artificial spectra, which can be searched through libraries. So as we did with the 4 normal modes and the PAcIFIC scan, we can look to see how MS2E plots in this view. As MS2E fragments all parent ion masses simultaneously and records all possible fragment masses, basically it covers the whole plane. But the fragmentation process isn't happening all the time. So the fragmentation mode cycles on and off for the duration of the LC run. And then fragment ions are correlated to their precursor ion based on their retention time and the shape of their chromatographic profile. So here are some things to think about following this session. Can you remember what the difference is between hard and soft ionization? can you list and describe the 4 main modes of tandem mass spectrometry? And can you describe the key features of DDA and DIA analyses and explain why DDA is data dependent but DIA is not? I've also made an HTML5 version of this video. The benefit of watching the presentation in HTML5 mode is that you get to go through it at your own pace and answer questions and problems as you go along to help you understand the material. To find the HTML5 versions, go to the Kilgour Lab website at www.kilgourlab.com and then go to the Teaching Resources page. On there you'll find a whole variety of resources to help you learn about mass spectrometry. I hope you enjoy them. So that brings us to the end of this session, which was a very basic introduction to tandem mass spectrometry.